Okay, good evening everyone. Good evening all of you. Good evening, good evening. Okay, let's join quickly. Sadhguru, there are total 30 ATPs dear. Okay. Because in the C4 cycle, Calvin cycle is also happening, right? In the Calvin cycle, we have already studied 18 ATPs. Where? In the bundle sheath cell. Okay. Whereas in the mesophyll cell, we will use the 12 ATPs. So total, there will be 30 molecules of ATP we will spend in the C4 pathway. You're getting my point? Total 30 ATP in the C4. That's correct. That's correct. That's correct. Okay. So friends, today we are going to finish the process of replication in both prokaryotes, and we will try to finish even in the eukaryotic organisms also. Okay. Everyone pay attention. DNA replication in the prokaryotes. So friends, so far we understand the rules and we understand the enzymes. Friends, now we are going to read the major process, that is process of DNA replication in prokaryotes as well as the eukaryotes. There is a no doubt when compared to the prokaryotic organism, the process of DNA replication in the eukaryotic organism is the most complicated process. We all know that very well. It's not an easy process to occur. Good evening, good evening. So friends, I am requesting all of you, give me 15 minutes first. Let me explain you every single point in a elaborative manner. Once I explain this particular 15 minutes, then I'll give time to ask all your doubts. So I'm requesting all of you just pay attention, utmost attention about every single point that we are going to discuss. So friends, DNA replication in prokaryotes, whether it is the process of replication, process of transcription, process of translation, doesn't matter. What are the three common steps, friends? The three common steps are the initiation, elongation, as well as, friends, termination. Don't do anything, just listen to me peacefully. You will get a crystal clear idea. Just pay attention. It seems to be very confused, that's why you should not divert your attention. Just pay utmost attention. Initiation, what is happening, friends? You know, right? During the, I'm just summarizing. During the process of initiation, two parent strands are separated. There will be a joining of the daughter strand here, correct na? So I mean to say, there will be addition of the primer, then elongation, the daughter strand can be synthesized. During the process of termination, it is going to stop. Now all of you listen. In the process of initiation, friends, two things will happen. What are the two things will happen? First, friends, good evening all, please continue. First, pre-replication complex will formed. What you can call pre-RC, pre-replicative complex. And in the initiation only, friends, the pre-replication complex is going to be converted into activated replication complex. one is the pre-replication complex, one is the activated replication complex. So now friends, all of you just listen to me very carefully. Let's talk about the initiation. And I have told you very clearly, in case of a prokaryotic organism, the DNA replication never ever start randomly. Everyone listen to me 15 minutes continuously. As we read that DNA replication is not happening randomly. The DNA replication always occurs at one particular point. The point where the replication will start, that particular point only we will be calling it as the ORC. We have read this also, friends. In case of a prokaryotic organisms, how many ORCs are present? One ORC only present, friends. One ORC consists of how many number of base pairs, friends? One ORC basically consists of 245 base pairs. Always remember that point. Friends, this is one ORC contain 245 base pair. Listen to me very carefully, friends. If you zoom that particular 245 base pairs, or if you zoom that particular ORC, you will get a different type of segments. All of you listen. 1, 2, 3, 4, 5. Here 5 segments are there. What do you call it, this particular 5 boxes? These 5 boxes we will be calling it as the DNA A boxes. Now you can ask me a question, sir, why we are calling DNA A boxes? If all of you quickly recall my words, I have told you DNA A is what protein? Initiator protein, which is basically starting the process of initiation. So my friends, during the process of initiation, this particular DNA A recognize the ORC exactly where? Exactly at this boxes. That's the reason we will be calling it as DNA A boxes. So there are 5 DNA A boxes are there. Each one is how many base pair? 9 base pair. Then friends, just next or upstream to the 9 DNA A boxes, there are another 3 boxes are there. Each one is how many base pair, friends? Each one is the 13 base pair. You know what do you call it, this particular 13 base pair, friends? You will be calling it as the DUE, DNA unwinding element. What do you call it, my friends? You will be calling this as the DNA unwinding element. Don't forget this. This is very important. I am just summarizing to all of you. I will explain especially molecular biology chapter 100% of depth. So pay attention, try to understand. If you can't connect one point, next point cannot be understood. So there is only one ORC is present. One ORC contains 5 DNA A boxes and 13 base pairs we can call DUE sequences, DNA unwinding element. Friends, if you look into this particular 13 base pairs, friends, they are extremely rich of adenine and thymine. Always remember this particular point. But friends, if you look into this particular DNA A boxes, I want all of you remember it contains, this is the sequence, and I don't want you to read and remember this particular sequence. It's not at all required only. Now friends, what are the major proteins that are involved? Everyone pay attention, friends. Of course, we require the DNA A protein. What did I tell you about the DNA A? DNA A is the one which recognizes the origin. Friends, everyone answer my question. Just remember, who is going to separate the double-stranded DNA? Obviously, DNA B. What we called DNA B? Helicase enzyme. But the question is very clear, right? How does DNA B know? How does helicase comes to know where is this particular ORC? Helicase comes to know with the help of DNA A. So always remember, DNA A recognize the initiator region first. Once it recognize the initiator region, then DNA B we already read, my friends, with the help of DNA C. What did I said about the DNA C? It is acting like a ATPase. So DNA C will provide the energy for the DNA B to open this particular double-stranded DNA. Very simple point. And we all commonly know, friends, SSB function is what I said a number of times. It will prevent the rejoining or reannealing. It prevent the degradation also. Very simple point. Then if there is any coiling occur, the gyrase or topoisomerase enzyme will come and remove that coiling. Everyone of you look into the diagram very carefully, don't get confused. It's all about confusion. The more briefly you understand the concept, more easily you can answer the questions. See here, this is the ORC. We already read. What does the ORC consist of, friends? ORC consists of a 9 mers. ORC consists of a 13 mers. Now you only try to understand DNA A protein. You are able to see here very clearly the green color one. Where does the DNA A will come and attaches to the 9 mers? Now all the students will get one doubt. What is the doubt you will get? Sir, you said that DNA A protein is the one which is starting the initiation, where the DNA A is associated to the 9 mer. That is absolutely true point. But the question comes here, if the DNA A associated with the 9 mer, DNA B also has to open the double stranded DNA from here itself, right? But it never happens. DNA B with the help of DNA C, from where it is start opening the double stranded DNA, friends? It starts opening from the 13 mers. Now you can ask me, sir, why it starts opening the DNA from the 13 mers? What is the reason? Because 13 mers contain A and T regions which are easy to break, which are easy to separate. That's why, listen my statements once again. DNA A recognizes the 9 mer, but DNA B starts opening the double stranded DNA from the 13 mers only. Now I want every one of you has to understand 2 points. The differences between the pre-primosome complex and primosome complex. Everyone pay attention, friends. Look into the diagram. Imagine there is a no primer. Still the primer was not there. My friends, now all of you look into this. What are all present now? DNA A is present, DNA B is present, DNA C is present, SSB proteins are present, gyrase or topoisomerase enzyme is present. Friends, in a very simple way, without primer, what my friends, without primer, what do you call it this as? You will be calling it as the pre-primosome complex. And friends, how does this particular pre-primosome complex is going to be converted into active primosome complex? Pre-primosome converted into active primosome complex by what, friends? By the addition of the primer. By the what, friends? By the addition of the primer. So upon who will add the primer? Of course we know very well, primase. So pre-primosome complex can be converted into the primosome complex just by the addition of the primer by the enzyme primase. Till here every student can understand very easily. My friends, it is very important to understand the regulation. If you get a toughest questions either from the replication or from the transcription, the toughest questions always come from the regulation. Even the prokaryotic DNA replication regulation also asked twice, every time for the part C only. Everyone pay attention. Friends, now initiation is over, is it? Because friends, we have added the primer. Now what we have to do? We have to elongate the strand. Now every student has to listen my statements, whatever I am saying. Friends, now after the initiation, elongation has to be occurred, then followed by the termination has to be occurred. Listen my statement. Before completing the elongation and before completing the termination, we should not start, listen to me carefully, we should not start another round of the replication. Now you can ask me, sir, how can you say another round of replication will start? You only try to understand, friends. All the initiation machinery are free. All the enzymes which are involved in the DNA replication initiation, all are extremely free. So there is a chance. What chance? Before completing the elongation and termination only, they will start immediately another round of initiation. So we have to stop that particular process. Now I can't stop every enzyme, right? But what I can do, I can block the ORC. Yes or no, friends? If I block the ORC, use the brain. I will repeat it twice, use the brain. If I block the ORC, the DNA replication initiation enzymes cannot be active. Hence, no new round of initiation will start before completing the elongation and termination. Very, very, very simple point it is. Now, all of you pay attention. How can I control the initiation? See friends, post ORC has to be blocked. What friends, what do you mean by the word post ORC? Already replication started, initiation is over. After that we can call it post ORC. Now this particular post ORC has to be blocked. It has to be blocked from what? It has to be blocked from the interacting with this particular initiation machinery. My friends, this process we will be widely calling it as sequestration. I will explain twice for all of you. Pay attention. What do you call sequestration? Blocking the post ORC not to interact with the initiation machinery, not to start new round of initiation immediately. All of you listen. How I am going to block the ORC? How I am going to block the ORC? With the help of an important inhibitor molecule. There are many inhibitor molecules are there, but one of the most important inhibitor molecule which can block the ORC, we can call it SEC-A. SEC-A is the inhibitor molecule which is actually blocking the post ORC. Let me tell you how. All of you pay attention towards the diagram, try to understand in a clear cut way. These two are the parent strand. We already read, right friends? The parent strand, every 1000 nucleotide they are having the palindromic sequences. This is the GATC. And we also know very well palindromic sequences at the adenine, they are getting methylated. Now we all know very well during the process of DNA replication, what is happening friends? During the process of DNA replication, the two strands of DNA get separated. Of course, true. Friends, once the two strands of DNA get separated, what is happening friends? There will be a synthesis of the daughter strand. Every student pay attention. Now my friends, there is no doubt, daughter strands got synthesized, no doubt. Now you only tell me, does the daughter strands also contain palindromic sequences? Now tell me friends, does the daughter strands got methylated? Not at all. Palindromic sequences which are present on the daughter strands, they are not methylated. When they are not methylated, we will be calling it as the hemimethylated DNA. My friends, when the hemimethylated DNA is there, immediately what is the inhibitor? SEC-A. Listen my statements carefully. Now what the SEC-A will do? SEC-A will go and block that particular ORC. Once it blocks the ORC, do you think another round of initiation will occur? Never. Immediately elongation termination occur. Now here comes a very interesting point. Now you can ask me, sir, how long you can call daughter DNA as the hemimethylated DNA? Don't you think hemimethylated DNA can be converted into methylated DNA? Of course. After certain time, what is that enzyme? Called as DAM, deoxyaminomethyl enzyme. What is that? Deoxyaminomethyl enzyme will come and add the methyl group even to this particular hemimethylated DNA also. After that, now you see here, daughter strand also become methylated. Now can you compare? Can you differentiate which is the parent strand and which is the daughter strand? Can you differentiate? You cannot. When both strands are in the methylated form, the SEC-A can immediately get inhibited. Immediately comes out. Here I am giving you one hint to all of you. SEC-A, which is an inhibitor, always associated with the daughter DNA friends in the hemimethylated form only. It is not going to be associated with the DNA when the DNA is a methylated form. So for all of you whomever did not understand, I am re-explaining the concept. Everyone pay attention and try to understand it a clear manner. What I am talking here, once the replication initiation is over, after the initiation is over, what has to be done? Elongation has to be done and termination has to be done. Before completion of elongation and termination, once again initiation should not start. So for that reason, what I have to do? For that reason, I have to block the ORC. When I block the ORC, what happen? Even though all the initiation enzymes are ready, they can't do anything, because if they want to do something, they have to start from ORC. But I blocked ORC. So that blocking of ORC only, friends, we will be calling it as the sequestration. How can I block the ORC? I can block the ORC by an inhibitor we called SEC-A. And what every student has to remember, if the SEC-A inhibitor want to attach us to the post ORC, first it should attach us to the DNA which is in the hemi-methylated form only. If something is in methylated form, friends, it never ever going to be associated. Now for that, you look into the diagram. These two are the parent strand. Parent strands contain the palindromic sequences. Palindromic sequences get methylated. During the replication, two parent strands are separated. Each strand acting as a template. There is a synthesis of the daughter strand. My friends, daughter strand also contains what? Palindromic sequences only. But my friends, do you think the palindromic sequences got methylated? Not at all. When the daughter strands are not methylated, what do you call them as? Hemi-methylated. After some time, they will become methylated. There is no doubt about it. Whenever they are in the methylated form, immediately the SEC-A will come and block the ORC, because SEC-A can only associated with the hemi-methylated form. So as a result, initiation is stopped. New round of initiation is stopped. After some time, what happens? There is an enzyme called DAM will go and add the methyl group to the hemi-methylated DNA also. Then SEC-A will get confused, which is the hemi-methylated DNA, which is the methylated DNA. SEC-A comes out. Once the SEC-A comes out, in meanwhile, process of elongation is also completed, process of termination also completed. Friends, this is the process of regulation of prokaryotic initiation. Friends, is that clear, everyone? Now all people can ask me the question. I'm good, Shivani. Thank you. Okay. Everyone say yes or no. Is that clear? I will explain for all of you if anyone having any doubt. I explain it twice. I hope you understand it. Is that clear, my friends? Everyone, quickly say yes or no. If you have any doubt, I can explain. Friends, I will explain everything very briefly. Just pay attention. That's all. You will understand. After listening my lesson, if you read the book, you will feel very easy. I hope it's clear for all. That's great. That's great. That's great. Good to know. Good to know. Others, come on, friends. Everyone respond. Oh, great. So, friends, now let's start about the process of elongation. So friends, when we are reading the elongation is common, and I hope every student actually remember elongation can be okay friends, please continue. Once again, I will explain for the period of 15 minutes and listen to me for 15 minutes very clearly. We will finish it off. Okay, everyone pay attention. The process of oh yeah, that's what, na dear, I am saying SEC-A is inhibitor, dear. What is the function of a inhibitor, dear? Inhibitor is the one which will go and inhibit something, right? So this is the ORC, see, this is the ORC. It will completely blocked by the SEC-A. If it got blocked, the ORC is not open. If the ORC is not open, the initiation machinery cannot do anything. So here the function of SEC-A is inhibitor, inhibiting something, blocking something. If it block the ORC, look at my hand, this is ORC actually. When the ORC is in the open form, enzymes can come. If the ORC in the closed form, enzymes can't attach us. What the SEC-A will do? SEC-A will block the ORC. I hope you got it. Right. Okay, good. Now friends, after the process of initiation, let's finish the elongation and termination together. Okay, now listen. The process of elongation. Friends, we already read. You should not think that there are two DNA polymerase 3 enzymes are coming and the two DNA polymerase 3 enzymes are doing the process. That is absolutely wrong statement. There is only one DNA polymerase 3. I have already told to all of you. it will undergo what? It will undergo the process of elongation. Sorry, it will only synthesize the leading strand also. It will only synthesize the lagging strand also. There is only one polymerase. But we read, I hope every student remember, in the diagram also we have seen, there are one polymerase having two subunit. Alpha epsilon theta 1 core unit, alpha epsilon theta 1 core unit. How these two are connected with each other? We already read with the help of tau dimer. So one core enzyme will hold leading strand and one parent strand, another will hold lagging strand and another parent strand. Very simple, my friends. Every one of you look into the diagram very carefully, my friends. This is the helicase enzyme, correct na? So the replication fork is moving like this, my friends. This is very important, friends. Focus. Replication fork is moving like this. Now I told to all of you very clearly, listen my statements once again. Whatever the parent strand that is moving towards the replication fork, listen, whatever the parent strand moving towards the replication fork, that is what exactly, friends, we will be calling it as the leading strand. So friends, this is what enzyme, friends? Helicase enzyme. Okay, now what is the point I want you people to remember? This is how helicase is moving. Everyone look into the diagram. The replication, along with the replication fork only, another strand is moving. So the strand which is moving exactly towards the replication fork, we call it as the leading strand. Every student has to remember, my friends, in the leading strand only one primer is going to get, one primer only will be there, one primer, and polymerase continuously elongating it. But my friends, in the lagging strand, all of you pay attention. See here, Okazaki, see, look into this, friends. One primer, Okazaki fragment, primer, Okazaki, primer, Okazaki, primer, Okazaki. But my friends, fork is moving like this, right? But the strand is moving opposite. That is the reason we call it as the lagging strand. But my friends, what is the common point every student has to remember? Whether it is a leading strand doesn't matter, whether it is a lagging strand doesn't matter. The daughter DNA is basically synthesizing in which direction, my friends? Obviously in the 5' to 3' direction only. There is no doubt about it. So what is the unique statement you have to remember? According to the rule of replication, whether the strand is moving towards the replication fork or away from the replication fork doesn't matter. Daughter strand has to be synthesized in the 5' to 3' direction only. Always remember that particular point. Now, friends, what is the length of this Okazaki fragments in case of a prokaryotic organism? I want all of you remember for the exam point of view, friends. While we are discussing the practice questions in the practice sessions, I want everyone to answer. Length of one Okazaki fragment is 1000 to 2000 nucleotide. Just one fragment only, friends, 1000 to 2000 nucleotides. Don't think that it's a small one. But what is the point? Like this, how many Okazaki fragments are present? I can tell like this. Sorry, like this, friends, thousands of Okazaki fragments are present. Thousands. Now, this is in prokaryotes. I want you to compare, friends. If you look into the eukaryotic organism, friends, what is the length of one Okazaki fragment? Length of one Okazaki fragment is just 100 to 200 nucleotide. That's all. The Okazaki fragment is a very small fragment in the eukaryote. So one fragment is just 100 nucleotide. But how many numbers? My friends, this is the unique statement. It is present millions in number. What, my friends? It is present in the millions in number. Do not forget the statement. So always length of the Okazaki fragments are more in prokaryotes but less in number. Length of the Okazaki fragments in eukaryotes are small but more in number. Okay, this is what elongation. In this particular slide also you can see very clearly, friends. Here whatever the arrow I am showing to all of you, this is the direction where the replication fork is moving. Now you can see this red color. This strand is moving exactly however the replication fork is moving. That's why I can call it as a leading strand. Now you see the another strand, it is just moving opposite. That's why we are calling it as the lagging strand. But what is the common step? Both are synthesizing in the 5' to 3' only. Now all of you listen to me very carefully. Till what extent, friends, all of you listen to me carefully, till what extent the DNA polymerase enzyme is moving continuously? Friends, until it reaches a particular point. What do you call that particular point, my friends? We call that particular point as the termination point. I will explain very briefly to all of you. I want every one of you pay attention. Look into the diagram. Just look into the diagram. I will explain in a very simple way. Okay, I am talking about the termination. See friends, this is the circular DNA, where in case of a prokaryotic organism. Friends, I have told you 3 to 4 times whether it is this is ORC. I told you 3 to 4 times whether it is a prokaryotic organism doesn't matter, whether it is a eukaryotic organism doesn't matter. What did I tell you? The type of replication always bidirectional replication only. That is what I told to all of you, if you quickly recall. Now same way in case of prokaryotes, this is the ORC. One replication fork is moving like this, one replication fork is moving like this. Everyone listen my statements, which means one replication fork is moving in the clockwise direction, another replication fork moving in the anti-clockwise direction. One is moving clockwise, one is moving anti-clockwise. Now every student has to remember, on the DNA there are certain termination sites are present. On one side 5 termination sites are present. 1, 2, 3, 4, 5 termination sites are present. On another side also 5 termination sites are present. 1, 2, 3, 4, 5. So on one side 5 termination sites are there. On another side also 5 termination sites are there. Now all of you please focus what exactly I am talking about. Everyone has to pay attention. Now this is the fork which is coming in the clockwise. Just listen. It is the fork which is coming in the clockwise. And friends, this is the fork which is coming in which direction? Anti-clockwise. All of you pay attention. Look into the concept. Understand the concept. Whatever the fork that is coming in the clockwise direction, see here, the termination sites are also in the same direction. Use the brain. See here, fork is coming like this. You only try to understand the arrow. The termination sites are also like this, like this, like this. They can easily cross. When the clockwise replication fork, when it comes here, do you think it will cross? Do you think it will cross these termination sites? Never ever. Now you look into this also. This is what a counterclockwise or anti-clockwise replication fork. The anti-clockwise replication fork also can cross this. Easily cross this. Cross these 5 termination sites and it will come here. Do you think it will cross these termination sites? Not at all. Now every student listen the words what I am saying. Listen my words. The replication fork which is moving in the clockwise direction, it will trap. Where it will trap? It will trap at the termination sites present at the anti-clockwise direction. Same way, whatever the replication fork that is moving in the anti-clockwise direction, it will trap the termination sites present at the clockwise direction. By that, the process of termination occur. You all people can look into this diagram. You will get little more crystal clear idea. This is one fork coming in clockwise. This is another fork coming in the counterclockwise or anti-clockwise. Now 5 termination sites are present in the clockwise. 5 termination sites are present in the anti-clockwise. Whatever the 5 termination sites are present in the clockwise, that can easily, very easily crossed by this fork. It crossed because they are all in the same direction. Now do you think, my friends, the clockwise fork will cross these 5? Never. So it will block here. It blocked here. Over. Now this fork, it can easily cross these 5. Do you think it will cross these 5? No, not at all. That's why I am using a word, my friends. Clockwise replication fork. Block at the termination sites present at the counterclockwise or opposite. Same way, counterclockwise replication fork can block at the termination sites which are present at the clockwise direction. Very simple. Now here comes another very interesting point, my friends. The interesting point, if this particular TER site, we can call it TER, TER site wants to be active, always it requires a protein. Without that particular protein, the termination never ever starts. What is the name of that particular protein? We called it as the TUS protein. TER protein, TUS protein. So TER and TUS together, they can form one complex. They will block the replication fork. This fork can be blocked here. This fork can be blocked here with the help of termination, TER sites and TUS protein. This is called, friends, the replication termination in prokaryotic organism. Now you can ask all the doubts. Is that clear, everyone? Friends, is it clear? Now you can ask all your doubts. I want every one of you to respond very fastly. Okay. Okay, I hope it's clear for all of you, my friends. That's great, that's great, that's great. Okay, now what we have to study? We have to, which is ATP independent? Can you ask me very clearly? See, dear Sumaya, this diagram, Sumaya, I understand your doubt. This diagram is not respect to the prokaryotes, okay? You remove this diagram, okay? It's wrong diagram. It's not at all ATP dependent. It's a ATP independent only, okay? Don't confuse. Okay, now let's talk about the process of replication in eukaryotes. I'm requesting follow me very regularly for the molecular biology. Once I finish the entire chapter, I'm giving you words. Once you read the concept will become 100% crystal clear to all of you. So only if you attend only replication and if you skip the transcription, if you attend the translation class, trust my words, you never get any concept. So you have to connect throughout the chapter, then the concept will be crystal clear. Okay, good. Okay, now the replication in eukaryotes. Friends, what are the fundamental differences between the replication in a prokaryotic organism and replication in the eukaryotic organism? I will tell you one simple line to all of you. What is that? In case of a replication in prokaryotic organism, friends, it is a very simple process. Very simple. Why? Because the genome of the prokaryotic organisms are very simple. But here it is what, friends? It's a complex process. It is a complex process. And the second point, what I want you people to remember here is that, everyone listen. In case of prokaryotes, how many ORC is present? Only just one ORC. But my friends, here instead of using the word ORC, what is the word we will use? We will use the word replicator. How many replicators are there, friends? Many replicators are there, my friends. Please don't confuse. I can tell an example, or minimum 150 replicators are present. Do not forget the point at all. So, and one more very important step, friends. When compared to the eukaryotic organism, the process of, this is really a good point. When compared to the eukaryotes, the process of DNA replication in the prokaryotic organism, it occurs very fast, very, very fast. But friends, in case of a eukaryotic organism, the process of replication is extremely slow. My friends, even though the process of DNA replication is a slow process, friends, it is exactly accurate process. Without any errors it will occur. The best technique to remember the replication, transcription, translation, gene regulation in eukaryotes is always try to compare with the prokaryotes. If you really compare with the prokaryotes, the process will be very easy. Let me explain you how. Now listen to me carefully. So let me explain you 15 minutes continuously. Replication in eukaryotes is the most complex process. replication fork moves very slowly, multiple origin, that is what do we call replicator. There are more than 150 replicators are present. My friends, here multiple enzymes are involved. Now you only tell me DNA A protein, that is called as what? Initiator protein. Yes or no? Of course yes. Don't confuse. This is what protein? Initiator protein in prokaryotes. Now in case of eukaryotes, this is where every student will confuse. The name of the initiator protein in case of a eukaryotes, we called ORC. This is the name of the protein, friends. You will be calling it as the origin recognition complex. That is the name of the protein. Don't confuse, which will go and associated with the replicator. DNA, what it has done in the prokaryotic organism, DNA A will go and attaches to the ORC. Here name of the protein only ORC in the eukaryotes, it will go and associated with the replicator. And in case of a prokaryotic organism, what we have used the versatile enzyme DNA B helicase. But my friends, the unique statement all the student has to remember here, in case of a eukaryotic organism, we don't use a particular or a specific helicase. We don't use. My friends, in case of a eukaryotic organism, the DNA polymerase alpha itself having helicase activity. This is a beautiful line every student has to remember. DNA polymerase alpha itself having intrinsic helicase activity. Do not forget that. And DNA polymerase delta. We read in case of a prokaryotic organism, the most important DNA polymerase is the polymerase 3. Very simple. In case of a eukaryotic organism, the polymerase delta is the most important enzyme. So friends, let's compare with the prokaryotes and let me explain to the eukaryotes. It will be very easy to all of you. Whatever I am explaining you in the diagram, the same point by point I was written here, so you can read it easily. Friends, if you want to understand the process of DNA replication initiation in the eukaryotes, every student keep it in your mind. The process of initiation will happen in 2 stages. First stage, we can call it as formation, formation of a pre-replication complex. Second step, my friends, activation of the replication complex. First step is what, friends? Formation of a pre-replication complex. Second step is activation of a replication complex. I want every student has to remember one more important line. What is that important line? Listen to me carefully. In case of a eukaryotic organism, let's take a human body. In our body only, the process of DNA replication is linked with the cell cycle. Yes or no? Of course, the process of DNA replication is linked with the cell cycle. Because friends, you all know very well in which phase the DNA replication occur. S phase is the phase where the DNA replication occur. But you all know very well, in case of a DNA replication, if the cell want to enter into the S phase, it can't enter directly. First, the cell has to prepare itself. So my friends, the question is very clear. When the cell is getting prepared, the cell is getting prepared in the G1 phase. All of you listen my statements very carefully. In the G1 phase, pre-replication complex will form. What is forming in the G1 phase? Pre-replication complex. Now you can ask me, sir, what do you mean by the word pre-replication complex? Assembly of all the enzyme, synthesis of all the enzyme, synthesis of primase, helicase, topoisomerase, DNA polymerase, everything all get assembled in the G1 phase. Now every student will get a doubt. Sir, you are saying that all the machinery which are required for the DNA replication, they are all synthesized in the G1 phase only. Then you will get a doubt. Sir, then why the DNA replication is not happening in the G1 phase? Because you said all are ready. Because my friends, all are ready. There is no doubt about it. But they are not activated. That's why we use a word pre-replication complex. The pre-replication complex wants to be converted into active replication complex. Where it will occur? It will occur in the S phase only. I will give you one more very best example. An example, I am having a bike. I am having a new bike. and I know driving also. But do you think I can drive without a proper license? I can't drive. So same way, even though cell prepared everything in the G1 phase itself, but the cell never undergo the DNA replication in the G1 phase, because even though it prepared everything, they are not activated. That is what do you call the pre-replication complex. Pre-replication complex occurred in the G1 phase, but my friends, pre-replication complex is going to be converted into the active replication complex only in the S phase. Very, very simple point it is. Now let me tell you what are the proteins that are involved. This is the DNA, and once again, without confusing, I am reminding to all of you, here's the name of the protein only, ORC, correct na? ORC, where it will come and attach. Suppose example, this is the reason where we called it as the replicator. So to this replicator, who will come and attach us? ORC will come and attach us. The moment ORC comes and attaches, immediately friends, ORC is going to recruit two important proteins, either CDC6 or CDT18. Most of the time, ORC recruit the CDC6 only. Sometimes even it recruits the CDC18 also. Then my friends, ORC is there, CDC6 or CDC18 also there. Now ORC and CDC6 together, they are going to recruit an important protein. Please pay attention. They are going to recruit an important protein we called CDT1. Now you only look into this. What are present here? ORC is present, CDC6 or 18 is present, CDT1 is present. These three proteins together, what my friends? These three proteins together, they are going to recruit what? They are going to recruit MCM. What do you call MCM? In case of a eukaryotic organism, MCM is called as helicase. What do you call it as? Helicase. Every student remember one important point. You can look into this. Helicase having how many subunits? 1, 2, 3, 4, 5, 6 subunits. Helicase will come and associate it. There is no doubt about it. Now you can ask me, sir, where all these are all happened? My friends, all these are all happened in the G1 phase. Simple question. All these are all happened in the G1 phase. Now, once again, you will get a doubt. Sir, here MCM is ready. Now, MCM is function is what? MCM is acting as a helicase. Function of a helicase, opening the double-stranded DNA. Now, it never opens. What is the reason? The reason MCM is not get activated. Now, you can ask me, sir, why the MCM is not activated? This is the interesting point. And this is what we got the 4-marker question from eukaryotes in the graphical way. If this particular MCM, how many subunits I told you? 6 subunits are there MCM. Among 6 subunit, 5 subunits of the MCM has to be phosphorylated. My friends, when the 5 subunits of MCM get phosphorylated, then only MCM will be active, then start opening the double-stranded DNA. But the question is very clear. If the 5 subunits of the MCM has to be phosphorylated, it requires an important protein. What is that protein we called? SCDK protein. Now you only tell me, does the SCDK is present in the G1 phase? Not at all. SCDK is present exclusively in the S phase only. Very simple and unique statement. So that's why even though MCM is assembled in the G1 phase, the replication never starts. That's why this entire complex we can call the pre-replication complex. Pre-replication complex is going to be converted into active replication complex only in the S phase. This is how we can regulate the process of initiation. Now, I want every student has to look into this diagram very carefully and try to understand the given concept. G1 phase, S phase, G2 phase, M phase. We all commonly know that. What did I reminded to all of you? I am writing 2 points. I want every student to understand the 2 points. In the G1 phase, pre-replication complex formed. In the G1 phase, pre-replication complex formed, but not activated. This is a very unique statement, but not activated. Then friends, look into the S phase. in the S phase, whatever the pre-replication complex that is already formed in the G1 phase, that is getting activated. Then in every student, remember, do you think new replication complex will be formed here? Not at all. No new replication complex will form. Very, very, very simple point. Now every student look into the given question carefully. Given graph, sorry, given picture. In the G1 phase, which levels are low, friends? CDK levels are extremely low. Which phase? G1 phase. As a result, what is happening? The pre-replication complex just formed. That's all. What do you mean by the pre-replication complex? Assembly of all. All the protein got assembled, but they did not get activated. Simple. What is happening in the S phase? CDK levels are extremely higher, my friends. In the S phase, CDK levels are high. Once the CDK levels are high, this pre-replication complex which is already formed, it just get activated. That's all. It just get activated. See here, it got activated and it opened the double-stranded DNA. But do you think once again new replication complex will formed in the S phase? It never ever happen. Because if the new replication complex wants to be formed, friends, the CDK level should be low, but in the S phase, CDK level will be high. So if you remember these two points, you can answer any graphical questions very easily. Now I am going to draw 4 graphs. I want every student has to answer that which graphs are correct, which graphs are wrong. Okay. Pay attention, all of you. Okay. Graph A, graph B, graph C, graph D. Okay. I am writing the concentration of the CDK. Okay. I am drawing concentration of the CDK in G1 phase as well as the S phase. Once again, I am writing what? Concentration of the CDK in the G1 phase and S phase. This is the G1 phase. It is same actually. Okay, G1 phase, and this is the S phase. This is the first graph. Now friends, all of you please kindly look into the second graph carefully. This is the second graph. Same concentration of CDK. And this is the G1 phase. Okay, concentration of CDK. And this is S phase. Same, no changes. G1 phase, S phase. Now I am drawing the third graph. Same, CDK, same. I am drawing G1 phase and I am drawing S phase. Same, G1 phase, S phase. The level of CDK. And I am drawing the fourth graph. Please look into the fourth graph. Pay attention everyone. This is the concentration of CDK in the G1 phase, and this is the concentration of CDK in the S phase. G1 phase, S phase. Now I want every one of you to answer. Among 4 graphs, which graph is a true graph, which graph is a false graph? What I want you to tell me, in which graph the concentration of CDK will be as per the normal terms. You know in which phase CDK should be high, which phase CDK should be low. Now you tell me which graph is right. Oh, I think you are unable to see na. 1 second. Just give me one moment of time. Just give me one moment of time. Now I think you can see. Now you can see. Now you can see. Now you people can see. Now tell me friends, which graphs are correct, which graphs are high, which graphs are wrong, which graphs are true, or which graph is false. I hope everyone can see the C and D also correctly. A, C, D are right, huh? Okay. Anyone else? All wrong. Okay. Everyone. Others. Friends, it's a right or wrong. Please give me the answers, please. Please give me the answers. Okay, what about other friends? See, this is the same question which was asked in the exam. Same part C question. See friends, all of you listen to me carefully. If you look into the level of the CDK in the G1 phase, it is the low. If you look into the level of the CDK in the S phase, it is extremely high. Now if you look into the 4 graphs, I have given 4 graphs wrongly. You tell me friends, in first graph, in the G1 phase and S phase, both the CDK concentration is same. It's a wrong graph. In the second graph, in the G1 phase, CDK level is high. So it's a wrong graph. In the third graph also I have given equally. Fourth graph also I have given equally. Correct na? Then all graphs are wrong graphs. Fantastic friends. This is the same question they have asked in the exam in the part C. Okay. This is what friends, the process of regulation of initiation in case of a eukaryotic organism. So in the next class we will finish this elongation and termination. We will move to the transcription. Okay, now everyone say yes or no friends. I hope everyone is clear till the process of initiation. Now you can ask all your doubts. Anyone having any doubt, I will explain. Please ask me. Okay. And friends, I am confirming that is today is Monday, tomorrow is Wednesday, Tuesday and Wednesday. Friends, that is from day after tomorrow. That is from Wednesday onwards, we will be having morning classes. Okay. So please kindly prepare for that. Okay. I hope it's clear for everyone friends. Okay. Okay. Great. That's all for now. From Wednesday onwards, we'll go little faster. Friends, don't worry at all. I will finish the syllabus 100% by May 31st. You don't worry about it. I'll go very faster. Okay. Okay, friends. Just be regular enough. Okay. I will see you tomorrow sharp 8:30 p.m. Bye. Thank you all of you. Good night.